Now we are going to see the Unit 1 continuity of Chapter 2 Kingdom Animalia in Zoology. So, before lesson we see the living world, how we are classify the animals, then how we are giving a name to the animals, what are the nomenclatures are there, all the things we are seen in a before lesson. The same basis we are seeing continuity we are seeing in this lesson. So the kingdom Animalia, there are millions of animals and they study with them without basic classification may lead to confusion. So that now we are seeing additionally what are the basic classification is occur. Then in Animalia kingdom, what are the non-chordates and chordates are there, and then the subphylums and also the classes, phyla are seen in this lesson. So now we go to basis of classification. So how we classify basically the animals, because in Animalia all are coming under multicellular cells. So in this multicellular, how we are classified the animals based on basic classification. So we are seeing four types of basic classification here. One is a levels of organization, means cellular level organization, then tissue level organization, organ level organization, and organ system level organization. So the levels of classification means all members of kingdom Animalia are metazoa and exhibit different patterns of cellular organization. So the cells of metazoas are not capable of independent extent and exhibit division of labor. So among the metazoa, cells may be functionally isolated or similar kinds of cell may group together to form a tissue or organ system. All the things are know that before a lesson itself. So next we go to cellular level organization. In the basic level of organization seen in a sponge, the cells of the sponges are arranged loosely aggregated and do not form a tissue. So they not gather and form a tissue. They are loosely arranged thing we are seen in a sponges. So in the cellular of organization, the cells, the different types of cells are functionally isolated. In sponges, outer layer formed a pinocyte. Pinocyte, that is a flat like a cells maintaining a size and structure of the sponge. The inner layer formed a canocytes. So they are flagellated collar cell maintaining water flow through the sponges. So respiration and digestion function. So this is a cellular level organization we seen in a example sponge. Tissue level organization, next level of organization. So the perform similar function are aggregated to form a tissue. So the cells of tissue integrated highly coordinate fashion perform common function due to the presence of nerve cells and the sensory cells. The tissue level organization exhibit diploplastic animals like Echinoria. The formation of tissue first step towards evolutionary body in an animal. So in a Hydra coelenterata, organ level organization and the animal will be and next level of organization, tissue level organization, they are moving. So next we go to organ level organization. So different kinds of tissue aggregate. First cells only doing a function. Example sponge. Tissue level organization, some cells aggregate to doing a function that is called tissue level. Organ level organization means different kind of tissues. They are arranged to form an organ and doing a specific function that is called organ level organization. These are evolution. So single cell to a few cells combine, the same like a cells combine to form a tissue and doing one function. So different type of cells are forming together and doing a function. It is a next level of evolutionary next level. So in this advanced tissue level organization appeared for the first time in phylum Platyhelminthes. So first level tissue level organization coelentratas, then organ level organization we seen in a phylum Platyhelminthes. So next organ system level organization, most efficient and highest level of organization among the animal exhibit by flatworms, nematodes, annelidans, arthropods, molluscs, echinodermids and chordates. These are next evolutionary that organ system are converted into organ. You know that what is organ, what is organ system, what is tissue. So in the first atoms are combined to form a molecule, molecules are combined to form a cell level organization, sponges. Then these cells are combined, same like a cells are combined to form a tissues. So this tissue level organization function will be seen in coelentrates. Then some different type of tissue combined to form an organ, heart, lung, that like organ. So that type of organization it is we are seeing in the platyhelminthes. Then organ, many organ combined to doing a one particular function, that is called as a organ system. So we are seeing this type of organ system organization next higher level, all that flatworms start to flatworm into chordates. So this evolutionary mesoderm in these animals lead to structural complex. The tissues organization form an organ and organ system. Each system is associated with a specific function, show a organ system level of organization. So in this platyhelminthes as only a single open, we are seeing these different. So in organ and organ system of organization, in the simple or complex depending upon individual animals, this organ system. So in a platyhelminthes, both opening, that is anus and mouth, are same or single opening only. Anus is also doing a function, mouth is also doing a function. So this type of thing is a incomplete organization, organ system organization. It is called as an incomplete digestive system. From Asclepianthes to chordates, that all animals having a complete digestive system. Two openings are in the body, anus and the mouth. Similarly, the circulatory system also modified in this organ system organization. So we see two types of circulatory system. One is open, another one is closed. What is open? Open means without any vessel, tissue space, the remaining filled in a blood will be filled in the tissue space. They are not having special capillaries or blood vessels. So where we see in this type of open type of organ system organization, arthropods, molluscs, echinodermids and urochordates. And closed type of blood vessels circulatory system where we seen in a that means in this closed type we are seen in annelidans, cephalopods and vertebrates. What is closed type means the blood is passed through the capillaries, arteries, veins and capillaries. Particular vessels its blood will be passing, so it is called as a closed type of circulatory system. So these are the difference between organization: cell, tissue, organ system. Organ system also some difference we are seeing. Based on this difference we are classifying the animal. So next we go to diploplastic and diploplastic organization. First we are we are taking one particular animal, we are classify that animal how they are having a cells or tissues or organs or organ system. Based on that we are classification. Next we go to this is a diploplastic animal or triploplastic animal. So what is diploplastic, what is triploplastic? So di that means it's two, tri that means three. So during embryonic development, the tissue and organs of the animal originate from two or three embryonic germ cells. Based on that, we are classify as a diploplastic animal or triploplastic animal. So, in this, based on the embryonic layer, external ectoderm, internal ectoderm, only the embryonic layer is having two layers, that ectoderm, external ectoderm and epiderm, that means ectoderm and internal endoderm means it is called as a diploplastic, or otherwise it give a rise to ectoderm is to give a rise to epiderms. So automatically endoderm, mesoderm, epiderm, gastroderm. So based on the gastroderm gut cavity is there. So based on that the body is divided. So they are called as a diploplastic animal. Example, corals, jellyfish, sea anemones. Animal which having developed embryo as the base, three germic layer is called as a triploblastic animals. So the outer ectoderm is forming a skin, having a skin, air, neurons, nails, teeth, etc. Inner means gut, lung, liver. So middle they are grown la muscle, bone, heart. So this is a diploplastic animal. So where we these type of a triploplastic animal means flatworms to chordate all are triploplastic animals. So we are seeing this diagram how the classification, germline classification are there. So in this germline classification, so the cell zygote will be developed as a germ. So in the germ only diploplastic animal means endoderm and ectoderm only presented. So in between the mesoglia is there, so diploplastic animal. In triploplastic animal means pink color we are seeing inside, white is that, it is called as a endoderm. So next, sorry, that is a coelom. Near a blue color is a light blue color is a this is a coelom fluid. So next this is a endoderm. The outer layer is called as a ectoderm. In between the pink color is that, the dark pink color cells are there, that is called as a mesoderm. So here we never see, lightly pink is there but is not having any cells. So there's a space is there, but in a triploplastic animal that space is also occupied by a cells. It is called as a mesosome. So triploplastic animals. Next we are undergo the classification based on the patterns of symmetry. What is called symmetry? So the symmetry is the body arrange which part of having a that lies on the opposite. When you are cutting any animal, the both sides are same, identical. They are having same organs in the other side also, that is called as a symmetry. That is called as a symmetry. So next how they are classifying based on the symmetry, the animals are classified. So they are, when you are cutting an axis in the animal, axis in a particular axis in animal, the opposites also identical. So they are called as a symmetry. So based on the symmetry we are classifying animal. So there are asymmetry. So asymmetry, radial symmetry and also biradial symmetry, all are seen here. First, what is asymmetry? So asymmetry means when you are cutting an animal in one particular axis, in one particular plane, they are not have an identical thing, that is called, they are irregular in shape, or we cannot cut in a particular plane, they are called as a asymmetrical. Example, sponges. We are seeing here some animal, we are cutting the animal, the shapes are not in regular. So when you are cutting in any axis, they not having a same identical, they not having two equal halves. So they are called as a asymmetry. Examples, sponges. Next, in adult gastropods, that means snails are also the same asymmetrical body plane. Then symmetrical animals having paired body parts arranged either sides of plane axis or through the central axis. So they are called as a, so they are two identical parts we are seeing in an animal in a central axis, we are dividing. So that is called as a radial symmetry. When you are cutting the animal here, when you are cutting the animal in any axis, not in a equal part. So these are called as a asymmetrical. They are not identical nature. So they are called as a asymmetrical. So in a symmetrical, is for in a principle of symmetry in diploplastic animal, around the axis we are seeing So Chitanuria such as sea anemone and corals are radially symmetrical. Our triploplastic animal like echinodermatous have a starfishes are a 5 plane of symmetrical, its own pentamerous radial symmetrical. Radial symmetrical are also in different ways. So it is a bilateral symmetrical and also pentamerous symmetrical are also we are seeing. Biradial means two identical. So there are two planes are symmetrical, two longitudinal as agitated axis, others longitudinal or transfer axis. Example, jellyfish. So they are bilateral symmetrical. So based on that symmetrical, we are classifying the animal. You are seeing radial symmetrical. When we are cutting a sea anemone, this is a sea anemone. When you are cutting a sea anemone in any axis, they are identical in nature. So this is a radial symmetrical. Pentamerous symmetrical means in a starfish. This is a starfish. When you are cutting a starfish in a pentamerous, so that is called as a pentamerous radial symmetrical. Biradial means in jellyfish, you are cutting a jellyfish in a axis. So they are having a central axis transfer, longitudinal transfer. Anyway you are cutting, it is having identical in nature, so it is called a biradial symmetrical. So in some insect also it's a bilateral symmetry. So based on the symmetrical nature, we are going to classify the animal. Next is coelom. Coelom means body fluid. The presence of a body cavity or a coelom, the important classification of most of the animals possess body cavity between the body wall and the alimentary canal, and it's lined with the mesoderm. So animal which do not having a cavity, body cavity, they are called as a acoelomate. You are using a before the coelom or symmetry. A means absent. So asymmetry means absence of a symmetrical. Acoelom means absence of a body cavity. So where we see this absence of coelom in body cavity in a Platyhelminthes. Next, in some animals, they are having a body cavity is not fully lined by a mesodermal epithelium, but mesoderm is formed scattered pouches between the ectoderm and endoderm. Such a body cavity is called as a pseudocoelom. Pseudo means false. So false coelom is there, false coelomic fluids are there. So they are called as a pseudocoelomates. So where we seen in a round worms, we are seeing the pseudocoelomate. Acoelomates or true coelom, the fluid is filled the cavity developed within a mesoderm lined by mesodermal epithelium called as peritoneum. Such animals are true coelomate or eocoelomates. So based on that mode of formation of coelom, the eocoelomates are classified into two types: schizocoelomates and also enterocoelomates. Schizocoelomates means they formed splitting of mesoderm. So annelid and arthropods, we are seeing the schizocoelomate. Next we are seeing in enterocoelomate animals, means body cavity formed from mesodermal pouches. So of a archipteron. So echinoderms, hemichordates and chordates, we are seeing enterocoelomates. These are the body cavity, coeloms, how it's present in a body. Next is segmentation and notochord. In some animal, body externally and internally divided into series, repeated units called as a segments. So these repeated segments that animals organs are undergo metamerism. The simple segments are found in annelidans, and each unit of body is very similar in next one. So seeing the segments, segments means already see some animals they are having a leeches. This is Platyhelminthes, we are seeing the segment. The same like segments are seen in some animals. So but in arthropods, the segments may look different and as a different function. In animals which are possess notochords, they are developed or called as a chordates. So backbones, the backbone development is started, that is called as a notochords. So the notochords in mesodermal dorsal sites during the embryonic development, some animals, based on the presence and absence of notochord, the animals are classified as a chordates. The chordate, cephalochordate, urochordate fishes to animals. So, and no, non-chordates means porifera to hemichordates. To hemichordate, they call it a non-chordate. Non-chordate means they not having a notochords. In a the presence of notochord, where it present, based on that we are classified cephalochordate, urochordate. So based on that we are classified that animal. So here we are stop this lesson. So in this lesson we started from the animal kingdom, how we want to classify the animal in organ cell, animal kingdom, basic level of organization in the cellular, or how they are a cellular organization, which animal undergo cellular organization, tissue organization, organ system organization, organ organization. Then next, how we are classified diploplastic, based on diploplastic and triploplastic. Then presence of coelom, then notochords, we are how we are classifying the animal, segments and notochords, how we are classifying animals. All the things we are seen in this kingdom Animalia. In this picture you are seeing, first we are all the multicellular animals are coming under to Animalia. Then cellular level organization, so we poriferas, all the poriferas coming cellular level organization. Tissue and organ system organization we are seeing. After that they are classified radial or bilateral, that is symmetry. Based on the symmetry, next we are undergo all the radials are not having symmetries, so they are under the cavities. So coelom, they are coelom trait and kidnopores are coming under it. Bilateral, we are classified. Next we are taken animal the cavities, coeloms, with the acoelomate, pseudocoelomate and coelomates. So acoelomate, platyhelminthes are being seen. Pseudocoelomate means ascalaminthus. Pseudo means false coelom, pouches are seen. So coelom means fully, the cavity is fully covered with the fluid. So annelidans, arthropods, mollusca, echinodermate, hemichordates and chordates are coming under the coelomates. So these method the animals are classified. Animal kingdom, fundamental classification of animal kingdom. In the next class we are continue the classification of Animalia. Thank you, students.